For much of the last century, the story of human origins seemed increasingly well-defined. Our species, Homo sapiens, emerged in Africa, roughly 300,000 years ago. Over time, we developed larger brains, more complex tools, symbolic behavior, and language. Then, beginning around 60 to 70,000 years ago, small groups of modern humans expanded beyond Africa, spreading across Eurasia and eventually to every inhabited continent. Along the way, we encountered other human groups, Denisovans farther east, but these populations were widely understood as evolutionary dead ends. They were seen as branches that diverged briefly, then disappeared, leaving us as the sole surviving human lineage. This framework shaped textbooks, museum exhibits, and documentaries. Fossil bones, stone tools, and archaeological layers seemed to support it. Each human species had its place on the map and its window in time. Neanderthals belonged to the Ice Age landscapes of Europe. Denisovans were a mysterious but limited presence in Asia. Other hominin species appeared older, more primitive, and clearly separated from the story of modern humans. The boundaries between us and them felt firm. Fossils are rare, often fragmented, and unevenly distributed. Entire regions of the world preserve almost no human remains at all. Archaeological cultures, defined by stone tools or artifacts, do not always correspond neatly to biological patrimony. Then quietly, a new source of information began to emerge. Not from bones shaped into displays or tools arranged in glass cases, but from something far smaller and far more intimate. Ancient DNA, genetic material preserved within fossil remains, offered a record that did not rely on outward appearance alone. It carried evidence of ancestry, population splits, migrations, and encounters that left no clear mark in stone or bone. As techniques improved and more ancient genomes were recovered, the genetic record began to speak with increasing clarity. It suggested that the world our ancestor inhabited was not shared with just a few familiar human relatives. Instead, it hinted at a far more crowded past, one in which multiple human populations overlapped, interacted, and sometimes vanished without leaving a clear trace in the fossil record. What followed would quietly but fundamentally alter how we understand who once shared this planet with us. Before ancient DNA transformed the field, knowledge of human evolution rested almost entirely on physical remains. Anthropologists compared skull shapes, tooth sizes, limb proportions, and brain case volumes to distinguish one human species from another. Archaeologists studied stone tools, hearths, and living sites to reconstruct behavior. From these lines of evidence, a structured picture of the past slowly emerged. In this picture, human evolution appeared mostly branching, but orderly. Early members of the genus Homo arose in Africa over 2 million years ago. Over time, some populations left the continent, giving rise to forms such as Homo erectus in Asia and later Homo heidelbergensis in Africa and Europe. From these ancestral groups, Neanderthals evolved in western Eurasia, adapting to cold climates with stocky bodies and large nasal cavities. In Africa, another population followed a different trajectory, eventually becoming an autonomally modern humans. This framework emphasized separation. Neanderthals were portrayed as distinct and isolated, living parallel to but apart from modern humans, until their extinction around 40,000 years ago. Denisovans, known initially from a single fingerbone and a few teeth, were treated as a rare offshoot confined to parts of Asia. Other archaic humans, identified through fragmentary fossils in places like Indonesia or China, were often placed deep in the past, assumed to have vanished long before Homo sapiens arrived. Migration was understood as a series of expansions and replacements. Wherever they went, earlier human populations were believed to fade away, either outcompacted or absorbed in ways that left little lasting impact. The prevailing assumption was that Homo sapiens succeeded largely alone, carrying a unique set of cognitive and cultural advantages. Fossils tend to preserve best in certain environments and rarely capture entire populations. Many regions that were crucial crossroads in human history Tropical forests, deserts, coastal plains offer few well-preserved remains. As a result, entire chapters of the human story were inferred rather than observed. Absence of evidence often became evidence of absence. Tool traditions were sometimes used as proxies for people. A change in stone technology was interpreted as the arrival of a new population or the disappearance of another. Yet tools can spread without people, and people can adopt new tools without genetic change. Even so, without another source of information, these assumptions filled the gaps. By the late 20th century, the dominant narrative felt stable. There were debates over details of exact dates, routes, and classifications, but the broad outline remained intact. Human evolution appeared to be a story of a few well-defined species, limited interaction, and a clear end time with modern humans standing alone. What few suspected was that this apparent clarity was partly an illusion, created by the boundaries of what could be seen. A deeper record existed, hidden within ancient bones themselves, waiting for the technology capable of reading it. The shift began at a scale invisible to the naked eye. Ancient DNA is fragile, breaking down quickly after death, under heat, moisture, and microbial activity. For decades, the idea of recovering genetic material from ancient human remains seemed realistic, making results unreliable, but gradual and viable. Researchers learned that dense bones, such as the inner ear region of the skull or the roots of teeth, could protect fragments of DNA for tens of thousands of years. In cold or stable environments, those fragments could survive even longer. Instead of looking for intact germs, scientists focused on recovering millions of tiny pieces, then using computational methods to reassemble them into usable genetic sequences. Once recovered, ancient DNA could be compared directly with the germs of living people and with other ancient samples. It allowed researchers to identify shared ancestry, estimate when populations split, and detect episodes of interbreeding, even when no clear physical trace remained. One of the earliest and most influential breakthroughs came from sequencing Neanderthal DNA. In the late 2000s, people living outside Africa carried small but measurable amounts of Neanderthal DNA. This was direct evidence that Neanderthals and modern humans had interbred after Homo sapiens left Africa. Soon after, a similar discovery followed from a remote cave in Siberia. A fragment of bone, too small to identify by shape alone, yielded DNA unlike that of either modern humans or Neanderthals. This population, later called Denisovans, was genetically distinct, yet closely related. More surprising genetic traces of Denisovans were found in present-day populations in parts of Asia. These findings alone reshaped human evolutionary history, but they were only the beginning. As more ancient genomes were sequenced from Europe, Asia, and Africa, researchers noticed something puzzling. Certain stretches of DNA in modern populations did not match known Neanderthal or Denisovan sequences. They appeared to come from other archaic human groups that had never been identified in the fossil record. In Africa, where DNA preservation is especially challenging, genetic studies of living populations revealed deep ancestral lineages that diverged hundreds of thousands of years ago. Some modern African groups carried genetic variants that could not be traced back to known fossils, suggesting interbreeding with ancient populations, whose remains have not yet been found or recognized in Asia. Similar patterns emerged. Ancient gums from sites dating to 40,000 years ago showed evidence of admixture with Denisovans, but also hints of contact with other, even more divergent human groups. These populations left genetic fingerprints without leaving clearly identified on the accumulation of genetic mutations over time. By measuring differences between genomes and calibrating them with known archaeological dates, researchers estimated when populations split and later reconnected. Many of these interactions occurred tens of thousands of years ago, during periods of rapid climate change and encountering one another across vast landscapes. What emerged from the genetic data was not a simple tree with clean branches, but a braided pattern of lineages. Human populations separated, met again, exchanged genes, and sometimes disappeared, leaving only faint traces in the DNA of their descendants. This genetic record revealed a world in which multiple human species coexisted, overlapped in time and space, and influenced one another in ways previously invisible. The past was not sparsely populated by a few isolated groups. It was shared, complex, and far more interconnected than earlier models had allowed. The deeper scientists looked into ancient DNA, the clearer it became that some human populations had left no obvious fossils behind. Yet they had still shaped the genetic heritage of people alive today, as ancient humans accumulated. A new picture of the human past began to take shape, one that challenged nearly every assumption formed from fossils alone. The genetic evidence revealed that modern humans did not emerge into an empty world dotted by only one or two distant relatives. Instead, our species entered landscapes already inhabited by multiple human populations, some closely related, others separated by hundreds of thousands of years. Genetic data showed that contact between populations was not rare or incidental. It happened repeatedly in different regions and over long spans of time. Neanderthal DNA entered the modern human gene pool shortly after Homo sapiens left Africa, likely in the Middle East. Denisovan DNA entered through multiple events, some affecting populations in Southeast Asia, others shaping the ancestry of people in Oceania. But beyond these known groups, the DNA pointed to others that remain unnamed. In Africa, the continent with the deepest human history, genetic studies revealed introgression from archaic populations that diverged from modern humans more than 500,000 years ago. These groups did not match Neanderthals or Denisovans. They represented deeply rooted African lineages that interbred with them. The evidence suggested that Africa itself was not home to a single uniform ancestral population, but a mosaic of groups separated by geography, climate, and time. Some lived in relative isolation, others interacted periodically. When modern humans' expopulation and genetic exchange followed, the result was a complex ancestry that cannot be traced to a single place or moment. In Eurasia, the pattern was equally intricate. Denisovans were not a single uniform population. Genetic differences between Denisovan DNA, found in Siberia, and Denisovan ancestry present in Southeast Asia, implied that multiple Denisovan groups spread across a vast region. Modern humans encountered these groups at different times, absorbing small but significant portions of their genetic material. Denisovans themselves had interbred with a hominin population that diverged from the human lineage nearly 1 million years ago. This meant that when modern humans encountered Denisovans, they were indirectly connecting with an even deeper bottlenecks. As modern humans spread across the globe, their populations repeatedly shrank and expanded. Small groups moved into new territories, carrying only a fraction of the genetic diversity of their ancestors. These bottlenecks amplified the genetic impact of interbreeding. Even limited contact with archaic populations could leave a lasting signal when human numbers were low. Some archaic genes appear to have been favored by natural selection. Variants inherited from Neanderthals and Denisovans contributed to immune responses. Skin biology and adaptation to high-altitude environments. These genes were not simply remnants of contact. They became integrated into the biology of modern population because they offered advantages in unfamiliar climates. Over generations, natural selection removed genetic variants that were incompatible. What remains today is only a small fraction of what was once exchanged. This loss explains why some ancient populations are almost invisible in the modern genome, detectable only through subtle statistical signals. Crucially, the genetic evidence. When a population disappeared as a distinct group, parts of its genetic legacy often survived within others. Some human species did not vanish completely. They dissolved into the populations they encountered. Their bones may be gone. Their tools unrecognized and forced a reevaluation of how human species are defined. Genetic boundaries proved to be porous. The idea of clean separations between species gave way to a model of long-term interaction, partial isolation, and repeated contact. Human evolution appeared less like a sequence of replacements and more like a network of populations sharing genes across time. The ancient DNA record revealed a past filled with encounters, some brief, some lasting generations, that reshaped human biology. Many of the people who once shared the world with our ancestors are known only through these genetic traces. They left no names, no myths, and often no identifiable fossils. Yet their presence is written quietly into the genomes of living humans. This understanding transformed lost human species from distant evolutionary curiosities into participants in a shared history, one that continues to shape who we are today, as the genetic evidence reshaped the map of ancient populations. It also changed how humanity itself is understood. An expansion of a family tree. It altered the meaning of what it means to be human, in a deep historical sense. Modern humans did not arise in isolation, nor did we move through the world alone. Our species developed within a wider human community, shaped by contact, exchange, and shared survival. Ancient DNA revealed that traits once assumed to be uniquely modern often have deeper origins. Some biological features present in people today were inherited from populations that no longer exist. As distinct groups, responses, adaptation, restaptistippen, restanak brizaps, resapri stempri, they are functional elements of modern biology. The discoveries also emphasized how interconnected human populations have always been. The boundaries drawn by geography or species labels were far more permeable than once believed. When different human groups met, they did not always compete until one vanished. In many cases, they formed families, exchanged knowledge. These interactions unfolded over generations, quietly shaping populations without leaving obvious archaeological signatures. This perspective shifts how extinction is understood. The disappearance of a population does not always mean total loss. Many ancient groups faded as distinct communities, but descendants live on, not as a separate people, but as part of the broader human population. In this way, human history is not defined solely by survival or failure, but by continuity through connection. Ancient DNA also reinforced the importance of diversity in human success. The genetic mixing between population introduced variation at moments when humans were expanding into new and challenging environments. It provided raw material for adaptation. When climates shifted, diseases spread, or food sources changed. Survival was not driven by a single perfect lineage, but by flexibility created through shared ancestry. These findings encouraged a more cautious approach to defining human groups. In the present, the deep past shows that human populations have always overlapped, blended, and reformed. Modern genetic differences are recent, shallow, and shaped by local history rather than deep biological divisions. For researchers, ancient DNA demanded humility. Long-standing models built on limited evidence, but they are no longer interpreted alone. Genetics provides an independent record, capable of revealing relationships invisible to the eye. When these lines of evidence are combined, a more complete and often more complex story emerges. For the broader understanding of humanity, the consequences are quietly profound. The people alive today carry within them traces of many others who once walked the same landscapes, faced the same uncertainties, and adapted to the same changing world. The human story is not one of replacement by superiority, but of persistence through connection. Ancient DNA does not erase differences between populations, past or present. Instead, it places those differences within a shared framework of deep ancestry. It shows that modern humans are not the end point of a linear process, but the living outcome of countless encounters across time. In revealing lost human species, ancient DNA ultimately reveals something equally important about ourselves. Our survival is rooted not in separation, but in shared inheritance, carried quietly forward, generation after generation. For much of human history, the past was reconstructed from what could be seen and touched. Bones, tools, and fragments of ancient landscapes offered glimpses of who came before us. Entire populations left little behind, and others were known only through incomplete remains. What ancient DNA has done, quietly and methodically, is expand the historical record beyond those limits. By reading genetic material preserved within ancient remains, researchers gained access to relations that never fossilized. Encounters that left no tools or bones could still be traced through shared ancestry. Human history emerged, not as a narrow path walked by one successful species, but as a broad terrain occupied by many human populations, whose lives overlapped in time and space, archaeology or anthropology. Instead, it strengthens them when DNA, fossils, and material culture are considered together. Each corrects and refines the others. Ancient DNA has shown that earlier models were not wrong so much as incomplete. They reflected the best interpretations possible with the evidence available at the time. This context. It explains why modern human diversity looks the way it does. It clarifies how populations adapted to new environments and survived periods of extreme change. It demonstrates that extinction in human evolution was often a process rather than an event. Most importantly, ancient DNA ghosts the continuity of human history. Many of the people who once shared the world with our ancestors did not vanish entirely. Their genetic contributions remain embedded within living populations, carried forward without names or memory. Their presence is subtle, but it is real as technique improve and new samples are recovered. Some lost populations may eventually be linked to fossils already discovered. Others may remain known only through DNA. Either way, the picture of the human past will become increasingly detailed, not by overturning evidence, but by adding depth to it. Ancient DNA reminds us that human history is larger than any single species. It is a shared inheritance, written not only in stone and soil, but within the biological record carried by every living person today.